Today we will be learning about molecular docking. It is a complex computational technique used in the field of bioinformatics and drug discovery. It is actually a method which analyzes the conformation and orientation of the molecules into the binding site of macromolecular target. So, in short, it is a direct interaction between a macromolecule and a micromolecule. So, a macromolecule could be any protein or any other macromolecules, and a micromolecule could be any ligand or the drug. So, in this technique, we can predict the binding modes and affinities of the ligand with the target protein. So, in this way, on computational mode, we can identify the reaction between a ligand and a protein. The major goal is identify potential drug candidate by understanding how they can interact with the target proteins. Now, the main principle over which molecular docking works is the energy calculated for different ligand poses, and the pose with the lowest energy is the more stable and most likely the binding pose. According to its principle, energy is being calculated. So, here we have an option in the software for energy calculation for different ligand poses in a sense. This is a macromolecule and this is a micromolecule or the ligand. So it goes and bind with different sites of the protein. So where the energy is low, at that site it would be forming the more stable binding pose. So, as I told you, for the purpose of molecular docking, we need certain softwares. And these are the softwares, that is Autodoc, Gram, Sanjeevani, GemDoc. So, Gram and Autodoc were developed in USA, and Sanjeevani is being developed in IIT Delhi in India. GemBank, its full form is Generic Evolutionary Method for Molecular Docking. So, these are the softwares where a selected ligand and a protein is being docked, that is being interacted, and their energy is being calculated over there. Further proceeding to its process, we have five major steps. That is the preparation of the structure of ligand and target protein. The first step involves ligand preparation. Second step is target protein preparation. Third is docking algorithm, scoring and ranking. And fifth is visualization and analysis. Now, let us look at each of this step one by one. The first one is ligand preparation. Now, a ligand can be downloaded from any software like PubChem, Zinc, etc. Now, ligand is prepared by optimizing its geometry and energy to ensure its conformation for docking. That is, a particular ligand with a perfect geometry and energy is being selected so that we could ensure its conformation while docking. Now, it contains methods like X-ray crystallography, NMR spectroscopy, and homologous modeling. That is, a ligand is being prepared with all these kind of methods so that a perfect ligand, a suitable and a ideal ligand is being made. Now, the second step is the target protein preparation. It is also a similar kind of step which involves the downloading of a 3D structure of a protein, which could be from any secondary databases like PDB and all. Now, receptor should be active. Receptor should be active means there are two steps, that is search space and scoring function. In space, it is a region where ligand is allowed to bind. Now, when we talk about the scoring function, it is used to calculate the energy of the different ligand poses. Now, the first one, which is the search space, in this, the ligand is allowed to bind. It is the region actually where the ligand could be binded. Now, when we talk about the scoring function, it is actually the energy calculation system. Now, after the selection of a ligand and a protein, the very next step is docking. Docking algorithm could be done in softwares like Gold, Autolock, Gram, etc. as mentioned before. Now, these are the softwares for docking purpose. Now, use scoring functions to calculate binding energy and affinity between ligand and protein. So as I told you, lower is the energy, more stable is the docking process. That is, more stable the interaction between the ligand and the protein is. Okay, so with this docking algorithm, energy is being calculated and the affinity between the ligand and protein is being estimated. The very next step is scoring and ranking. So, according to the energy binding sites and the kind of energy being produced by the interaction of a ligand and protein, we rank them accordingly. Then, the fifth step is visualization and analysis, in which the whole process of docking is being analyzed and a perfect docked ligand and a protein is being taken. Now, these are the applications for molecular drug molecular docking. The first step is identifying potential drug candidates. It is cost effective, target identification, virtual screening, polypharmacology can predict adverse drug reactions, ligand target binding, rationalization. So, without even going to practical experience, we can visually analyze what kind of reaction could be produced between a ligand and a protein. So, these are the applications of molecular docking. So, that's all about molecular docking. Thank you.